Hi, I'm Joshua Lee, and in today's video, we will be learning about protein synthesis. As the name suggests, protein synthesis is the process by which protein is made. I highly recommend watching my previous video because it'll make it easier to understand this video. So how is protein made? There are two main steps to protein synthesis. The first would be transcription. This is where your body copies the genetic information in your DNA and turns it into a message. Since DNA is stored in the nucleus of your cells, protein synthesis begins in the nucleus. The section of DNA that will be transcribed is called the transcription unit. An enzyme known as RNA polymerase copies the information inside the DNA and turns it into mRNA. The M stands for messenger. It does this by reading the nitrogenous bases in the DNA, adenine, thymine, cytosine, and guanine, and finding the complementary base for each one. If you remember from the previous video, RNA doesn't have thymine, so it's replaced with uracil. The RNA polymerase keeps forming this RNA until it hits a point in the DNA called a terminal signal. The terminal signal tells the RNA polymerase to stop copying information. Then we have the strand of mRNA. On one end, a special type of guanine known as a 5' cap is added. On the other end, a bunch of adenines known as a poly-A tail is added. These caps protect the mRNA and make it easier for it to attach to other organelles later on. However, this is not the end of transcription. Right before the mRNA leaves the nucleus, it goes through a process of editing called RNA splicing. This process is basically the editing of the RNA and removing any excess information that is not needed. After that, the mRNA is taken out of the nucleus. So now we open the DNA, copied the information into mRNA, removed the excess information, and carried it out of the nucleus. Now it's time for step 2, translation. After the mRNA leaves the nucleus, it goes to the rough ER. The little bumpy things on the rough ER are known as ribosomes, so the mRNA that left the nucleus goes to one of the ribosomes, which is made of rRNA. The R stands for ribosomal. In the ribosome, the mRNA gets attached to tRNAs. The T stands for transfer. tRNAs have an amino acid on one end and a 3-letter code on the other end. As I mentioned in the previous video, amino acids are the building blocks of proteins. When the mRNA goes through the ribosome reads the mRNA's nitrogenous bases 3 at a time. These sets of 3 bases are known as codons. The ribosome reads these codons and finds the tRNA with a matching anticodon, which is made up of the complementary bases. So if the first codon was AUG, the matching anticodon would be UAC, which codes for methionine. Most proteins start with methionine, so it is typically known as a start codon. Let's say the next codon is CCA. Then the matching anticodon would be GGU, which brings a proline amino acid. This new amino acid gets linked to the methionine with a peptide bond. The tRNA goes away eventually but leaves behind the amino acid, starting a polypeptide chain. On the topic of amino acids, there are much more codon combinations than there are amino acids. So there could be multiple codons that all code for the same amino acid. This is incredibly handy when there's a mistake somewhere along the transcription process and the codon happens to be wrong. Even if there is an error, it might not necessarily affect the final product. This is a chart used to find out what amino acid a tRNA would be carrying. These are the first base, and these are the second bases, and these letters are the third bases. So to use the chart, you need to first know the codon on the mRNA. Let's say the codon starts with A. Then the second base is U. Then the third base is G. We can find out which amino acid this will give us by connecting all of these. So we can see that it will give us a start codon, which is known as methionine. Let's say the next codon is C, C, and A. If we connect all these, we know that we'll get the amino acid proline. So there can be multiple codons that all code for the same amino acid. For example, UUA and UUG all code for leucine. In fact, there's 6 codons that all code for leucine. When using this chart, it is important to remember that you should only use this for the codons on the mRNA. This is not for the anticodons. This process continues with the ribosomes reading the codons and finding the tRNAs with the matching anticodons and continuously making the polypeptide chain. After all the codons have been read, we are left with a polypeptide chain. Sometimes this chain can be a protein in of itself, but most of the time it has to go through multiple stages of folding. The actual structure of a polypeptide chain before it goes through any folding is known as its primary structure. It's just a long chain of amino acids. After the first stage of folding, some hydrogen bonds form between the amino acids. Then, the primary structure becomes shapes like helices and pleated sheets, which is the secondary structure. Remember how in the last video we learned about R groups? Some R groups are hydrophobic and try to stay away from water, which can bend the polypeptide chain even further. Now we have a super complex 3D shape, which is its tertiary structure. And for its last stage of folding, sometimes multiple protein chains come together like hemoglobin and form a quaternary structure. In today's video, we learned about the process of protein synthesis where nucleic acids are used to make proteins. I hope you enjoyed this video. Please subscribe and thanks for watching.